Hi everyone, today's my topic for discussion is free radicals and antioxidants. Several types of reactive species are generated in body as a result of metabolic reactions in the form of free radicals. These free radicals can be either an oxygen derived or nitrogen derived species. They are usually called pro-oxidants. These pro-oxidants can attack macromolecules in our body, including protein, DNA, lipid, etc., and can lead to cellular or tissue damage. But fortunately, our body is having a specific mechanism known as antioxidant to counter their effect. Our body is gifted with antioxidants. These are produced either endogenously or it can be received from exogenous sources, and which include so many other types of like enzyme, minerals, vitamins, etc. Now, what are these free radicals? The definition for free radicals, these are oxygen containing molecules that have one or more unpaired electrons, which making it highly reactive with other molecules. They react quickly with nearest stable molecule to capture electron in need to gain stability. Okay, this figure which explains the nature of free radical. Free radicals can initiate chain reactions by extracting an electron from a neighboring molecule to complete its own orbit. But usually in our body, either through or an endogenous or an exogenous oxidant system, antioxidant system is there, which will helps to fulfill this nature, the grabbing nature of free radical, and helps to donate an electron from the antioxidant molecule. The types of free radicals, there are mainly three types of free radicals. First one, reactive oxygen species. Second one, reactive nitrogen species. And the third category is which will be an intermediate or reactive metabolites of various reactions like metabolic activation of drugs, toxins, pollutants, cigarette smokes, etc. So these are the examples for various free radicals. Superoxide anion, hydroperoxide, hydroxyl radical, hydrogen peroxide, singlet oxygen, nitric oxide, peroxynitrate. So in all these figures we can understand, we can find out a common thing that most of the free radicals are represented with a superscript dot, except the hydrogen peroxide and singlet oxygen. These are actually, even though it does not have a superscript dot, these are classified under the free radicals because of their extreme reactivity, which will be included under the free radical classification. Now, the actual definition of reactive oxygen species. Molecular oxygen, usually which will be completely reduced to water via oxidation reactions. But the partial reduction of oxygen which creates a highly reactive species, they are known as the reactive oxygen species. And the characteristic features of this reactive oxygen species are they have a short lifespan. Usually their lifespan will be milliseconds or very few minutes, something like that. For example, superoxide having half-life at 37 degrees Celsius 10 raised to minus 6 seconds, and whereas the hydrogen peroxide having half-life as few minutes. And the second characteristic feature is that high reactivity. Once a free radical form, it can initiate chain reaction. That is meaning of high reactivity. Then it can generate new reactive oxygen species by chain reaction. And the last one, which causes damages to various tissues. These are again given the examples of reactive oxygen species: superoxide anion, peroxide molecule, hydroxyl ion, so and so. Now this figure which shows the sequential reduction of oxygen, and as I have said already, the partial reduction of oxygen which is responsible for the production of these free radicals like superoxide anion, hydrogen peroxide, hydroxyl radical, etc. The partial reduction which leads to the generation of free radicals in human body. Now next, what causes free radical? What are the sources of free radical synthesis in human body? So the first one, cellular metabolism, which will be explaining the endogenous sources of synthesis of free radicals in human body. And the second category, which is represented in green box, under the green box, the second category is environmental factors, or which is also known as the exogenous factors, which is responsible for the production of free radicals in human body. So under the endogenous or cellular metabolism, the causes are electron transport chain, certain enzymes, oxidase enzymes, lipid peroxidation, nitric oxide production, phagocytosis, synthesis of prostaglandin, auto-oxidation of metal ions, flavin coenzymes, etc. So in all these metabolic reactions which involving these processes will be responsible for the synthesis of free radicals in human body. Let's discuss one by one later. Now the second category, exogenous sources of production of free radicals or environmental factors, ionizing radiation, photolysis of oxygen by light, photoexcitation of organic molecules, cigarette smoking, air pollutants, drug metabolism, alcohol, etc. Now the first one, electron transport chain. So ETC, we know that constantly produced during normal oxidation of foodstuffs. ETC is the final oxidative pathway for the complete oxidation of foodstuffs where the energy is being generated. So due to leaks in the electron transport chain in mitochondria, it can be leads to the generation of reactive oxygen species. So about 1 to 4% of oxygen taken up in the body is converted to free radicals. Mitochondrial reactive oxygen species production is modulated largely by the rate of electron flow through respiratory chain complexes. So this is the first way of production of free radicals in human body through the electron transport chain. So when the electron flow occurs through these complexes, there will be escape, or for partial reduction of oxygen occurs, which will be leading to the synthesis of reactive oxygen. And the second enzyme is xanthine oxidase. Xanthine oxidase, recently we have learned that, which is responsible for the synthesis of uric acid in human body, or which is the enzyme which catalyzes the catabolism of purine bases, and finally the production of uric acid occurs. So this product, during the production of uric acid, there will be generation of this superoxide free radical occurs by the enzymatic reaction of xanthine oxidase. Now let's see what is the role of phagocytosis in generation of free radicals. Phagocytic cells such as monocytes, macrophages, neutrophils, eosinophils, etc. will be responsible for the formation of large quantities of free radical, especially superoxide, then hydrogen peroxide, hypochlorous acid, etc. and will be responsible for the destruction and removal of foreign cells. So during phagocytosis of foreign particles, specifically bacteria and all, the phagocytic cells are activated and they exhibit a rapid increase in oxygen consumption. That is known as a phenomenon known as a respiratory burst. So this respiratory burst is responsible for the destruction of bacteria. Here in this figure, the NADPH oxidase, which is located in the plasma membrane, is responsible for the respiratory burst. NADPH oxidase is inactive in resting phagocytic cells. which will be activated upon contact with various ligands with receptors in the plasma membrane. So here during phagocytosis, NADPH oxidase which converts molecular oxygen from the surrounding tissue into superoxide. So that is the first reaction of phagocytosis. NADPH oxidase converts oxygen from the surrounding medium into superoxide, O2 superscript dot superoxide. In the second reaction, this superoxide is converted into hydrogen peroxide by another enzyme, superoxide dismutase enzyme. Next, in presence of myeloperoxidase enzyme, another enzyme which is a lysosomal enzyme which is present within the phagolysosome, which causes the conversion of this hydrogen peroxide into hypochlorous acid in presence of chloride ions. And this hypochlorous acid is responsible for the killing of bacteria. Excess hydrogen peroxide. Here you can see the excess hydrogen peroxide, which is either neutralized by catalase enzyme or the glutathione peroxidase enzyme, the glutathione peroxidase enzyme system, which consists of multiple enzymes, glutathione peroxidase, glutathione reductase, and glucose 6-phosphate dehydrogenase enzyme. These three will be responsible for the inactivation of the excess hydrogen peroxide, which will be synthesized during phagocytosis. So in this phagocytosis, we can see that the multiple free radicals will be generated as superoxide, hydrogen peroxide, hypochlorous acid, etc. So here the generation occurs, but at the same time it is having a positive effect, or which is mainly useful here to kill the bacteria. A specific disease where there will be genetic deficiency of NADPH oxidase system, which leads to a specific disease known as chronic granulomatosis. This is actually a disease characterized by persistent and multiple infections of the skin, lungs, bone, liver, and lymphocytes. Here in CGD or chronic granulomatosis disease, the NADPH oxidase will be completely absent in macrophages and neutrophils. So what will happen? So that will be, this enzyme is mainly responsible for killing of bacteria, but certain bacteria like streptococci, pneumococci, etc., that is having an ability to produce hydrogen peroxide by itself. Therefore, they are able to destroy the myeloperoxidase system by the macrophages. But another category of bacteria, specifically Staph aureus, Staphylococcus, being catalase positive, can detoxify this hydrogen peroxide in the macrophages and therefore are not destroyed in such persons or the person having this CGD disease. So what will happen? There will be recurrent pyogenic infections by Staphylococci are more common in chronic granulomatous diseases. Next is the free radical nitric oxide, which is produced in various cells in mammalian system by a reaction catalyzed by the nitric oxide synthase enzyme. This enzyme is responsible for the production of nitrous oxide, act as an intracellular signaling molecule and performs many biological functions in human body. And this nitric oxide itself is a free radical and can react with the superoxide to yield peroxynitrate, which decomposes to form the highly reactive hydroxyl radical. So this is the next mechanism to synthesize another free radical nitric oxide in human body from the amino acid arginine by nitric oxide synthase enzyme system, which is responsible for the production of the free radical nitric oxide. Next is, this is another mechanism of free radical production in human body, that is lipid peroxidation. This slide will be explaining later in the coming slides. Now, the other endogenous sources like prostaglandin synthesis. The reactions which is catalyzed by cyclooxygenase and lipooxygenase, which leads to the formation of hydroxyl and peroxyl radicals. Phagocytic cells, which we have already been explained. Then cytochrome P450 enzyme complex, which is found in endoplasmic reticulum and responsible for the hydroxylation of a wide range of endogenous and exogenous molecules, and which is also responsible for the generation of superoxide as an intermediate. So like these are the other endogenous sources: infection, ischemia, inflammation, autooxidation of phenylalanine, reduced riboflavin, immune cell activation, mental stress, cancer, aging. All these conditions are responsible for the production of various molecules of reactive oxygen and nitrogen species in human body. Now the exogenous causes of formation of reactive oxygen species. The first one is that ionizing radiation, specifically X-rays and ultraviolet rays, which can lyse water and leading to the formation of hydroxide radicals. Then another like cigarette smoking. Cigarette smoke, which contains many oxidants and free radicals and organic compounds such as superoxide and nitric oxide, and that is again a dangerous thing which will be responsible for the formation of free radicals in human body. Next is ozone exposure, which can cause lipid peroxidation and induce influx of neutrophils into the airway epithelium. So like this, these are the external sources for formation of free radicals. So here gives a picture of an apple. The left side is which is exposed to air, and the right side which is when the right side of apple is dipped in orange juice. So what does we understand that? That is the presence of antioxidants which causes the inactivation of the effect of free radicals. So we can understand that without antioxidant, it will be very dangerous condition, or it causes multiple deleterious effect in human body. So just for the symbolization which have been given this, where it have been given this figure, where the presence of antioxidants which can reduce the deleterious effect. Okay, now let us see in detail about the effects of reactive oxygen species. Oxygen radicals can damage to DNA, proteins and lipids in cell membranes and plasma lipoproteins. So the first one, it can cause the oxidative damage to PUFA in cell membranes and plasma lipoproteins, which leads to the formation of lipid peroxides. These lipid peroxides are highly reactive dialdehydes that can chemically modify the proteins and nucleic acid bases. And next is that oxidation of enzyme proteins, which leads to loss of function and fragmentation of proteins. Specifically, oxidative damage to the tyrosine residues in proteins can lead to the formation of dihydroxyphenylalanine that can undergo non-enzymatic reactions, which leading to the formation of oxygen radicals. The next is that this reactive oxygen species can cause damage in the DNA double strand, or it can cause the breakage of the DNA double strand. So indirectly, all these effects can cause the inhibition of protein or enzyme synthesis in human body, and which can indirectly leads to cell death, mutation, carcinogenesis, aging. So many things can be caused by the effect of reactive oxygen species. So here gives the picture in detail about the cells and tissue damage by reactive oxygen species. So lipid peroxidation, then degeneration or deactivation of the protein and enzymes. All these things will be leading to the injury of cell membrane and cell components such as like protein, PUFA, etc. So all these can leads to this kind of activity can leads to myocardial infarction, cerebral apoplexy, arteriosclerosis, etc. And that can be even leading to aging also. And DNA damage or mutation of DNA specifically, which is done with the double-stranded DNA strand breaking, can also cause mutation and cancer. Now let's move in detail about the lipid peroxidation. So lipid peroxidation is done with 3 stages, like initiation phase, second one propagation phase, and the third one termination phase. Specifically, PUFA, polyunsaturated fatty acid, which is present in the cell membrane, are easily destroyed by peroxidation. So now what is this lipid peroxidation? So here, during initiation phase, the primary event is production of carbon-centered radical, which is represented by a simple capital R with a superscript dot. That is known as carbon-centered radical or PUFA radical, or lipid peroxide radical, R double O with a superscript, that is known as lipid peroxide radical. This happens by the interaction of PUFA molecule with free radicals generated by other means. So many means, so many mechanisms we have seen where the generation of free radical occurs. Any such free radical will contact with PUFA, which is present in the cell membrane, will be responsible for the production of lipid peroxides. That is the initiation of the lipid peroxidation. And the second, propagation phase. The carbon-centered radical, which immediately reacts with molecular oxygen, and leads to the production of again a lipid peroxide. And this lipid peroxide will react with another molecule and the reaction will be continuing. That is known as the propagation phase. So here you can see that once a free radical generates another free radical in the neighboring molecule, it will cause a chain reaction which will go and bind with another molecule and so on. So new and new free radicals will be generated continuously. So chain reaction or propagation is initiated. So this is sometimes called a death kiss by free radical. That means which will be killing another molecule. Okay, so that is the second stage, propagation stage or phase. Now the last is termination phase, where the reaction will be continuing until a peroxyl radical reacts with another peroxyl radical to form inactive product. That is known as the termination phase. So this is explaining about the lipid peroxidation, which is more dangerous, which can cause so many deleterious effects to the human body. Next deleterious effect, damage to the DNA. Mainly mitochondrial DNA are more vulnerable to get damaged by the free radicals, and it is more commonly affected by the hydroxyl radical. Remember, mitochondrial DNA is more vulnerable and which will be more affected by hydroxyl free radical. It mainly causes DNA double strand breakage will be happening and directly cause inhibition of protein and enzyme synthesis. So the outcome of this DNA damage will be, you can see, if it is repaired by DNA repair mechanism, it can be leading to viable cell. But if this DNA damage caused by the free radical is not repaired on time, or it is leading to misrepair, which will be leading to either misrepairment will be leading to mutation and cancer, and if it is not repaired, which will be leading to the cell death. So this is all about the initial part of free radicals. We have seen what is the definition, what are the causes for the formation of free radicals. We have seen so many examples, and what are the deleterious effects of free radicals. Now in the coming session, we will be discussing in detail about what is the oxidative stress and what is the role of antioxidant to detoxify the free radicals. Thank you all.